Okay, this is our next DNA video. It's going to go in a little bit more detail. We're going to start talking about some genes and chromosomes, or how that DNA is organized in our body. So while we're doing it, the essential question is going to be, how do genes tell us how we are related to other organisms? So focus on that question as you're taking your C notes and as we're going through this. Purpose of it is to reinforce your knowledge of DNA, to identify what genes are, and to learn how DNA is organized in our body. So a real quick review of DNA. So if you don't remember, DNA is what codes for the RNA in our body, and that RNA ends up making the proteins. The proteins do just about everything in your body and make you pretty unique and make you different from each other and from different organisms. So if you remember, DNA is made into a double helix type structure. So it has two sides to it with the rungs in the middle. It's like that ladder. When you unfold it, it's going to look like it is over here on the left. You're going to have strands of DNA that look just like this, with a backbone, which will be this side, and the rungs of the ladder, which go across. So our DNA structure is going to be made up of nucleotides. And if you remember what a nucleotide is, it's just that sugar, or the penthouse, with the phosphate group, or the swimming pool, and one of 4 different nitrogenous bases. So every nucleotide has that basic shape, but there's going to be 4 different types. And if you remember, there's specific base pairing. So anytime I have an A for adenine, so that base there is an A, it's going to pair with a T. So A always pairs with T. So on the right side here, you can see I have another nucleotide now. I still have my phosphate group, my sugar, and my nitrogenous base, and that A has now paired with a T. One thing I want you to notice is that they are going in opposite directions. So the ones on this side are going to go that way. The ones on this side are going to go up that page. So I could start building a strand of DNA by adding additional nucleotides. So here's another nucleotide. I have my phosphate group, my sugar, and my nitrogenous base. This base happens to be a cytosine, or a C. And that C is going to pair with another one, a G. So here's another nucleotide. There's my phosphate group, my sugar, and my nitrogenous base, what happens to be a G. So C is always going to pair with G. And you can see now that I'm forming that backbone, and I'm forming that backbone with my phosphate groups forming a bond with the sugar from the nucleotide above it. And I could keep on building that strand of DNA, A to T, C to G, and this sequence of DNA is A, C, and remember that DNA would keep going, and I could have millions of nucleotides in that strand of DNA. So I could have millions of A, C's, T's, and G's all arranged in a different order. And what they're going to be arranged in are going to be our genes. So here's a strand of DNA. So here's a chromosome where my DNA was all wound up. If I unwind that chromosome, I have something called chromatin, but on that chromatin I have genes. So this highlighted portion is going to be a gene, and that specific sequence of A, T, C's, and G's are going to code for a protein. That protein is going to make you unique. So this gene might be the gene for hair color or eye color, and that sequence of A, T, C's, and G's might be a little different between you and your neighbors. One thing that's important to notice is that there's going to be a whole bunch of different genes on this strand of DNA. So there might be a gene here, maybe it's for eye color, or maybe a gene here that tells you if your hair is going to be straight or curly, maybe a gene here that tells you if you could roll your tongue or not. So anything that happens in your body is going to be determined by those genes, and those genes are arranged in your DNA. So a gene is just a portion of your DNA that codes for a specific trait or characteristic. So here is some DNA. So this is a chromosome. So when your DNA is all nice and packed together, it's going to be in this little bit of a shape here. But you notice that there are some colored bands here. And these colored bands, we're going to say, are going to be where your genes are on that chromosome. So this green band will be for eye color. This red band might be for blood type. Hair color could be the purple bands. And the orange ones here could be how tall you get. And it's important to know that there are approximately 30,000 known genes in humans. So humans have about 30,000 that we know of. Other organisms could have more or less depending on what they are and how much DNA they have. So here's just an example of one gene. This gene happens to be 522 nucleotides long. So if you count it, you're going to count 522 A, T, C's, and G's. Now most of our genes are going to be a lot longer than 522. They're going to be in the thousands. This is a short one, but this is going to be one gene, and it would be found on a sequence of DNA, and that gene will code for a specific protein. So let's talk about chromosomes. So chromosomes are how our DNA is arranged inside of our cells, kind of. So this would be a little picture of a chromosome. Here's a picture of a chromosome. You're going to see these pictures a lot. What you are seeing here is your DNA. So over on the left, you can see that double helix structure inside of there. That is representative of the DNA. Now on the right, it shows you kind of pulling it out like a line of thread. So this is our actual strand of DNA, and you can see the double helix shape with the cross members in here, which would be the bases. And this is how our DNA normally is. It's going to be all stretched out, and it's called chromatin. So chromatin is just going to be DNA that's kind of unwound. It's going to look like a bowl of noodles. And because it's unwound, those A, T, C's, and G's are exposed, and you're able to make an RNA, and that RNA can make a protein. When it's all wound up like this over here, this chromosome, those A, T, C's, and G's are kind of hidden, so it's really hard to turn that DNA into RNA and turn that RNA into protein. So when it's pacted or compacted into this chromosome structure, it's only going to be when the cell is dividing or replicating. So here's a little comparison of chromosomes versus chromatin. Once again, chromosomes are the DNA that are really tightly packed, and then chromatin is going to be the unwound DNA. So in this picture inside this nucleus, if you look really closely, you can see some purple lines. That's going to be the unwound DNA. And that's how it's normally found, because when it's unwound, we can make that RNA and we can make proteins. When it's all tightly packed in a chromosome, you're not going to be able to make as many proteins. So once again, DNA is usually unwound in the form of chromatin. So you could look in most of these pictures, you can't see any definite chromosomes, but what you can see are the dark spots of DNA, and that's just because the DNA is unwound in the form of chromatin. In some of them, you could start seeing DNA start to get formed, especially in this one. So these long strands here are actually DNA strands. This is our chromosomes. It's all compacted, and that's why you're able to see them under this microscope. And if you didn't pick up on it, this is going to be a microscopic view of some sort of plant. I know it's a plant because of the shape. It's got a nice structured shape, which means it's going to have a cell wall, which it's most likely going to be a plant. So let's talk real quick about a karyotype. So a karyotype is going to be a picture of all of our DNA. So you can see here that I have 1 chromosome, 2 chromosomes, 3, 4, 5, 6. If you count them all up, you're going to have a total of 46 chromosomes in a human body. They're going to be in 23 pairs. So this one has 22 pairs and then 1 sex pair. So this is a human karyotype because I have 23 pairs of chromosomes, meaning I have 46 chromosomes. And it's either going to tell me if it's a female or if it's a male. So on our sex chromosomes, if it's an XX, it's going to be a female. And if it's an XY, it's going to be a male. So if you see the 2 different length chromosomes, it's going to be a male because it has 1 X and 1 Y chromosome. The reason that women have more chromosomes, the X is bigger, is because they have more stuff going on in their body. They reproduce babies, so they have a different anatomy, and they have to do some really complicated chemical reactions inside their body to make that baby grow. Here's another karyotype. This karyotype only has 16 pairs, so I know it's not a human, and I know by doing some math that it has 32 chromosomes. So if I count them up, 1, 2, 3, 4, 5, 6, I would count up 32 chromosomes in 16 pairs. One thing I didn't mention in the other videos, these chromosomes, one comes from your mom, one comes from your dad. So they come together when that baby is formed, so like a sperm and egg come together and they form one cell that has a complete set of chromosomes. So my sperm cell might have 16 chromosomes. That egg would have 16 chromosomes. When they come together, I'll have a total of 32. That happens in humans too. So human sperm cell is going to have 23. Human egg cell will have 23. When they fuse together and that baby is being made, you will end up having 46 total chromosomes. Now, this is a little comparison of DNA strands. So this gene in species A matches exactly the gene in species B. So they're going to be really similar creatures. For example, it might be one species of a monkey and a separate or another species of a monkey. They have really similar DNA, but they're going to be different species because if you kept going on, there would be some differences later on down the road that make their DNA different. Most of this DNA is pretty similar, so you can see all of them have GACT at the end. All of them start with AC. I still start to see some differences in here, and because I have these differences, I'm going to have different species. So the further away I get from species A and species B, especially down here, this might be completely different type of organism than the monkeys that were in A and B, but we know by looking at their DNA that at one time they were closely related. So we'll go over this more after the semester break, but there's a common ancestor here. So species E, F, G, and H a long time ago had a common ancestor. Then you could go back even further in time and H through C had a common ancestor. And if you go even further back, all of them had a common ancestor back here. So what happened was there was some sort of mutation or evolution happened and that organism changed. And then whatever organism it became changed. And eventually we ended up with the species that we know about, which exists today. So that's a real quick overview of DNA and genes and chromosomes. If you have any questions, please come see me. Also need to work on your Quizlets and start working on your group projects. Thanks.